Once again, welcome everybody to the AIMM Portugal webinar, number 4 in our series. First of all, I'd just like to really quickly introduce who AIMM is, or who AIMM are. I know that some of you will have heard this 3 times already in previous webinars, so I apologize to all of you, but it's important that everyone knows who we are. So AIMM was founded in 2010. It's a not-for-profit NGO. We're based here in Portugal with the headquarters in Lisbon, but our field station is here in the south of Portugal in the Algarve region. It was really created by a group of young Portuguese marine biologists with the intention of creating a sort of research association to study the marine species and the marine environment of the Portuguese Atlantic Ocean. We're based here, you can see in the map, in the south of Portugal, in the southwest of the Iberian Peninsula. So this gives us quite an interesting location here on the sort of northeast Atlantic Ocean. The map on the right-hand side here gives you a bit more detail. We're based just outside Albufeira here, and so that's our area of research. So as I say, our field station is here in the Algarve, and this is where we operate our Dolphin internship program. So every year we receive interns and students from all over the world come to join us to learn about whales and dolphins and to actually study these animals in the wild. Now, obviously, this year's things are a little bit complicated, as you all know, but we have just started our internship season on the 1st of July, so we're about a week in already, and we have our first intern here with us already. So the internship program is open, it's up and running. We have all of the sort of health and safety, clean and safe measures in place that are enabling us to run our internship for the summer season more or less as normal. So I'll talk a little bit more about that right at the end, but let's get going and introduce our guest speaker. We have a very special webinar today. So we have a guest speaker, Laura van Zonneveld. She's an ex-research assistant of AIMM. So she worked with us in 2017 and 2018. So we're really pleased to have her back with us, albeit via Zoom, to tell us a little bit about her master's thesis project. So she's going to talk a little bit about that, but just very quickly to introduce Laura, she's now a lecturer in animal physiology, behaviour and ecology at Aarhus University of Applied Sciences. I probably didn't pronounce that correctly, I'm sorry. But anyway, she's going to be talking a little bit more detail about her master's thesis that she completed in 2015 in applied ethology and animal biology. But I don't want to give too much away, so I'll invite Laura to unmute her microphone. Laura?
Yes. Hello.
Yay, you're
it's
there.
Unmuted, yes.
Okay, what I'm going to do now is just change a setting here at Zoom so that Laura can share her screen with us. So Laura, hopefully...
Yep, I can see I am the host now. Now I need to share my screen. Yep. Hope this is all okay. I think you guys should be able to see my presentation now. If not, please shout.
I think so. I think it's just thinking about it for a second. I think there's a slight
it's just
delay
loading a little. Oh, it could be.
Zoom,
yeah.
I think. So
okay.
Just keep that in mind when you're switching through the slides. Okay, we can see your presentation.
Oh, perfect. Also, a little mini warning. I have a lot of pets in the house here. I have 2 dogs, I have 2 cats, and they tend to ask for my attention once in a while. So there might be a cat walking past the screen. or a dog noise in the distance. So just to warn you guys. Okay, so well, Henry already introduced me a little bit, but well, I also made an introduction about myself. So my name is Laura, and I did my master's thesis in ethology, and I'm also a teacher. So my bachelor's was in biology, general biology, and animal welfare. And I did a side program to become a teacher as well. And I was the research assistant with AIMM in the season of 2017-2018, which I really, really enjoy. And I'm still, yeah, I'm an ex-research assistant, but I'm still really involved with AIMM because they're a great organization. And actually, one of my students at the moment is writing her thesis on some data from AIMM at the moment. So at the moment, I'm teaching human and animal welfare and also behavior at Erasmus University. Henry actually pronounced it quite correctly, in the Netherlands, and they're based in Almere near Amsterdam. And yeah, well, that's what I do now full time. So to give you a little introduction on animal welfare, when we look at animals in wild animals in captivity, the scope of different institutions has changed quite a bit over time. Whereas before, a zoo would put more focus on the display of the animal, just pure the display. Nowadays, most zoos, the better zoos, I would say, are focusing a little bit more on education, research, conservation and welfare of the animal species that they keep. And to do so, they put a lot of time and effort into encouraging natural behaviors of the animals. Because if you encourage them to use their natural behaviors, of course, education of the public is a lot more interesting, because if you just see an animal sitting, not doing anything, you're not really going to learn much from it. So they try to make the enclosures in such a way where it looks natural, and also, of course, they try to encourage the animals to display their natural behavior so the public can learn from that as well. Which also gives a really good opportunity for research, because if you want to research this behavior, it's quite useful if they actually perform this behavior as well. So for example, there's a zoo near here, and they have like a fake termite hill for chimpanzees, which is really fun for the chimpanzees, really educated for the people to see how they use tools, for example. But there's also a lot of research done how this tool use is learned by the younger chimps, for example. Another part which is important is conservation. If you have the idea of one day bringing these animal species back into the wild from a zoo setting, which happens sometimes, it's quite useful if they still perform their natural behaviors, otherwise they might not survive. So that's why I also want to encourage these behaviors. And then welfare. We now know that it's really important for a lot of animals to display their natural behaviors in order for them to have better welfare, for them to feel better, and to not display abnormal behaviors, or at least try not to see that as much as some animals do. So some examples. Here you see like a big cat. Big cats, of course, if you look at natural behaviors, most of their time is spent, well, they sleep a lot as well when they've just eaten, but their active time is spent on foraging, hunting, and then eating a prey, which often takes quite a lot of time. They have a big carcass. Also, they're going around their territory, and they might fight off some conspecifics that they don't want in the territory at that moment. So a lot of natural behaviors that if you don't encourage those behaviors, if you just give a big cat a silver platter with some nice chunks of meat on it once a day, and they eat it, and that's all they do for the entire day, they start pacing around the enclosure because they have all this energy that they cannot spend on natural behavior, so they start pacing, start showing this abnormal stereotypy type behavior. So if you encourage these natural behaviors, you can do this in a way where you make the enclosure as much as a natural environment as possible. So for an animal like this, you can add rock structures, you can add trees where they can climb in, but also a place where they can have their own time if they don't want to constantly be viewed by people, because these animals normally live alone. They live in very quiet areas where they're not constantly in a lot of noise and with a lot of people. So you have to give them these natural parts to really keep them happy. And then for food, you can give them something, well, in this case, they made a sort of fake Nemo kind of thing, but you could also give them an entire horse carcass, for example, or hang it up somewhere or hide it somewhere in their cage so they have to forage like they would do in normal nature and take time to also consume a meal like that, which can take quite a lot of time. And other animals like lemurs, for example, you can use the entire space that you have. You can go up into the air with the space. You can hang up different structures. So they have to explore, they have to climb, have to find different foods. And in that way, you can encourage their natural behaviors. Another example, which you see more and more lately, is by putting different species together. So they can also enrich each other. Of course, in nature, animals are constantly around, well, their own species, of course, which is really good if they're social species, but also with other species. They learn from them, they interact with them. So if you have that opportunity to bring two species together that don't kill each other, of course, then that could be a really good enrichment as well. So if you look at these animals, a lot of opportunities to encourage these natural behaviors by using environmental enrichments to from the natural look of the enclosure to the type of foods that you give them. Another example of a lion that has to catch its food from a high beam. But when we look at dolphins, that's a little bit different, because dolphins, if you look at their natural behaviors, they also forage, they also hunt, they socialize with each other, and they socialize with different species. Also, they're very intelligent, they're very curious, so they scan around different environments, they're exploring a lot, all these types of behaviors. But when you look at a typical Dolphinaria type enclosure like this one, it doesn't really encourage these natural behaviors. And it's really difficult to do so as well, because you can't really make them hunt, because you cannot put a fish in chlorine type water. Also, you're not allowed to put a prey animal with a predator. So you can give them toys or something that they can play with. But these toys always float on top of the water. Or you can give them something heavier, for example, which will be laying in the bottom of the pool. So there's not much for them to explore. There's not much for them to swim around and to really explore and to do. So these dolphins often they get a lot of training, etc. But really, like performing natural behaviors, that's a little bit difficult. The type of dolphins that we normally see in captivity are the bottlenose dolphins. You all know them probably. Flipper is one of the most famous ones. Is the ones where it kind of started for dolphins to be seen more in captivity. A few facts on these dolphins: they're an echolocating species, so that's a very important natural behavior for them to be echolocating. They're very social. They live in groups of 4 to 30 normally in a type of fission fusion type group, which means that it's not always a stuck group, so they could be with different individuals depending on the day or their mood or whatever. Also, they're very highly intelligent. They're spending most of their time during the day being social, but also, as I said earlier, foraging and traveling, and they could even travel up to distances of 128 kilometers a day, but that depends a bit on the group and the type of food source that they live on. But if they want to, they could travel that much. So their sonar, the echolocating behavior provides their main sensory input. So we use our vision, which is our main sensory input. They use echolocation to explore the environment, for navigation, for hunting, and exploration of new areas. And they even use the sounds for some social aspects as well. So all natural behaviors that are really difficult to encourage in an unnatural setting. So this was a little part on echolocation. I had a little video, but unfortunately you cannot hear the sound, so I'll just play it and I'll just explain a little bit. Has some really cool visuals though. So this is a dolphin. So the beam that you see here, it shows what the echolocation would look like if you would be able to see it. So they produce these clicks in the back of their head by these phonic lips that vibrate together by air passing through. They produce these clicks. It goes really fast. It's like a buzzing sound when we hear it. And they pass through the fatty melon, which is this bump inside their head. That's why they have this bumpy head. and it passes through there so that it can create a beam, so they can actually emit this beam towards where they want it to be. So that's in short how it works. They have an entire structure inside, but I'm not going to go too much into anatomy. So when we look at echolocation, we want to research this a little further. Between these clicks, there's always a lag time. There's always a short space of time before the next click is emitted. And this is called the interclick interval, which typically can range between 20 to 40 milliseconds. So that's really, really fast. That's why for us it sounds a little bit like a buzz when you hear the clicks all together. When they are closer to an object, the interclick interval becomes shorter, of course, because there's more clicks emitted in a shorter time. So it can even be up to 2 to 3 milliseconds when they're going after prey or they're really close to a certain object. Of course, when they're further away, such as 20 meters, it can be up to 50 milliseconds. So when you look at this interclick interval, by the way, the sound in the background is one of the dogs playing with something. I hope it doesn't disturb you too much. Either way, when we look at this interclick interval, the interesting thing is that you can analyze these type of structures, or these type of click trains, we call them. And they are seen when a certain behavior is shown by the dolphins. For example, when they're scanning an area, it can be either a constant type click train where the interclick interval is the same every time there's a click emitted. That's normally they use that when they're scanning an area and they are emitting clicks. But it can also be kind of going up and down, longer interclick intervals or shorter interclick intervals. When they're approaching something, first the interclick interval is long and it becomes shorter the closer they get to an object. So you can kind of see them locking onto something and then they go to it. I'll show you later a couple of those click trains that we analyzed during this research. But we also saw a buzz quite often, and a buzz is when clicks are emitted below 16 milliseconds. And you can actually see the picture, you can see it here, a constant buzz below 10 milliseconds even. The interclick interval is really short, so it really sounds like bzzz, like that, like a proper buzzing sound. That's why we call it like that. So echolocation is a natural behavior for a dolphin, but it's also partially a learned behavior. So as I just explained why natural behaviors are so important for a zoo to constantly stimulate them to use it, it's really important for dolphins, of course, to still be using their echolocation just as they do in nature, in the wild. So the idea of this study, when you look at a dolphin pool in a dolphin area or a typical one, the environment can be quite barren. And this is because there has to be a constant flow of water. Water needs to be filtered constantly. You can actually see in these pictures, these little holes here, they are vents that where the water passes through. So if, for example, you'd put sand at the bottom or like a type of rock structure, you can imagine that the water cannot circulate properly, and then the hygiene would be below what you want it to be. So the dolphins can actually get sick. That's why the water column is often very empty and barren. But when you want a dolphin to use echolocation more, it can be a quite understimulating environment. So we want to see if we can make a type of underwater structure where we can acoustically enrich the dolphins, and possibly they could use these structures as landmarks as well. So I did my master's in Sweden, and the trainers there of the Dolphinarium, they asked us if we could see if making rock structures in the pool would not just look cool for visitors, but also be cool for dolphins, and how long they would really find that interesting. So how useful would it be to really make these rock structures in their environment? So we did this by employing these things, and I'm going to tell you a little bit more about that later. So first, what we needed were dolphins, and in the dolphin area there, at the time I was there, they had 9 Atlantic bottlenose dolphins. The ages were ranging between 1.5 to 41 years old. The water depth, so they have 3 pools, like a big sort of display pool, and they have a holding pool in the middle, and then they have the show pool. So we just looked at the display pool and the holding pool, because of course in the show you're not really going to get natural behaviors. So this is what it looked like. This is not one of my videos, it's just something I found on YouTube. So you can actually see the shallow area, which is 3 meters in the back. Then you see a slope, and then on the left of this video, there's actually the deeper area, which is 6 meters in depth. And this is where these dolphins were living in. And then, of course, the other pools as well. So the devices we used were 10 CPODs, and CPODs are short for continuous purpose detectors, and 2 PCLs, which is short for purpose click logger. And these devices are used a lot in conservation because they're normally put into the sea and picked up a month or even a few months later to see what type of activity is recorded. And they can see how many, if there's porpoise in the area, how many porpoise are in the area, and what are they doing? Because looking at the click trains, you can kind of see, are they maybe scanning the area? Are they hunting? Are they, I don't know. So they're used a lot for this type of research. And the PCL, the black one, you can see here, there was actually a new device at that time, and they also wanted to test it. So that was kind of a side project to my main thesis project. So how do these things work? Well, first of all, I just told you about how echolocation works. So these devices, they don't actually record audio, they record the amount of clicks. So every click emitted by the dolphin is recorded with this thing. There's a little SD card inside. There's also air inside, so they float a little bit. There's batteries inside, and that's about it. And then, of course, the recording devices. And when you take the SD card out, which we did once a week, you get a lot of numbers. You get a massive database with numbers, because I just told you the echolocation clicks is a lot of them in a short amount of time, and that's what it records. So you get big Excel databases, and then you can actually put them in the CPOD program, which is not very user-friendly because only researchers use them. And then you get graphs like this, which you can later analyze, and I'll show you some of the results that we got. So what we did was we put 10 of the CPODs, those are the blue dots, in the big area, 1 in the little holding pool, and then we put 2 PCLs in the middle of those. The PCLs we added later because they still needed to be programmed. So the first things we put in the pool were the CPODs. And this is how we did this. I'm not the one diving, I'm the one in the little boat over here, unfortunately. So there's bolts in the bottom of the pool, and we put the CPODs every week. I extracted the data, and we put them back in exactly the same place. So I'm actually in the boat with a map, and then telling the divers, okay, this CPOD needs to be there, this one needs to be there, etc. So every week we got the same data from the same area, so it didn't get mixed up. But that all sounds really easy, which it kind of was. Then we had 2 cameras. We had one roof camera, which we put on top of the roof, and it had a big lens, which you could actually see almost the entire pool. And we had one camera that showed the bottom of the pool, the lower part, and it was behind a window. And they were actually continuously recording for 12 hours a day, because after 12 hours it was too dark to see anything, and we did that for a total of 5 months. So this is what it looked like from the roof camera. So you can see kind of the entire display pool, and I made a grid to also record how much time the dolphins spend in each area, so we could see which area they preferred and what they were doing exactly. And this is what it looked like from the below camera, the one that was behind the glass. You can actually see the CPODs floating there, and you can see one of the younger dolphins, Perla, playing with the CPODs. She's sort of smashing them around, smashing them on the floor. She was one of the youngest in the group, so she was the most playful of all the dolphins. Okay, so that all sounds like it went flawless, which of course experiments and research never really goes flawless or actually expected. So we spent a lot of time designing these experiments, and then you put all the stuff in, and then everything breaks. So the first thing that happened was that the rooftop camera fell into the pool in week 1. We managed to save it by asking a dolphin to take it from the pool bottom and giving it to the trainers, and then we fixed the entire thing by putting it into a bathtub and then putting it all back together. So yeah, that was a bit shitty. Another thing that happened was that the sea pods were on a setting where you're supposed to record one porpoise in the entire North Sea, but now it was put into a pool with 10 dolphins and a concrete wall around it, so it was way too noisy to analyze anything from it in the first week. So we had to change the settings to a lot lower. Then when that finally worked, Perla, the young dolphin you saw in the video, kept smashing the sea pods against the floor. So that was a really funny game, which actually made the SD card lock break. We found out a week later that, again, all the data was gone because she broke all the SD card locks. So then I had to put that all back together. And then I was in the boat, and with the map, telling the divers where to put the sea pods, and the dolphins were so happy, and they kept throwing this ball in my face and like splashing me with water, and the entire map was destroyed. So then we put the sea pods in the wrong places. And then when that finally worked, because I laminated the sheets and I used different paper, we found out that the time of the devices, the sea pods, didn't actually match up with the time in the videos, which made it impossible to relate the video with the click data that we found. So all these things went wrong, but that's what you do with experiments. You fix it. So we made these little floaty devices. We made these discs so that the sea pods wouldn't crash against the floor as much. We used a lot of tape to put everything back together and to keep it together. And I started using waterproof paper. So what we eventually looked at, because then we had all this stuff ready to record for 5 months, and then you start thinking, okay, what can we actually record? First of all, what the trainers found most important, and the zoo, of course, was how long are these underwater devices interesting for the dolphins? So how are they enriching their lives? How can we encourage their echolocation? Is that working? So I used the data from the sea pods, of course, to see how much echolocation was actually produced. And I looked at the camera, the videos, to see what type of behaviors they were performing towards the devices. Then we also wanted to see how they used the area of the pool. Then we looked at the numbered areas, how much time they spend in each area. We wanted to see if they use the sea pods as orientation devices for when they're resting or when it's dark or whatever. And we also wanted to see what their diel patterns were. So what type of activity do they have? When are they most active, and what time of the day do they use most echolocation? So what we found, quite interesting things. So when we put the sea pods in at first, well, as I just told you, we didn't actually get much data from the first week. So that was really a pity, because we wanted to see if the echolocation activity increased, but we couldn't really record that in the first week, unfortunately. But I do have behavioral data, and we found out that after 3 weeks, or after 1 week, actually, they already stopped playing with the devices. Only the younger dolphin was still playing with the devices, which you can see here, like the touch behavior, and also looking at the devices. But the older dolphins, after even a few days, they didn't really care anymore. So that actually declined really quick. Then we added the PCLs, the black loggers. And then we wanted to see their behaviors towards it. So then we can actually use the echolocation data to see if their echolocation clicks were higher than they were before. So we actually found that in the first weeks, on average, they had around 20,000 clicks per hour per day and logger that we recorded. So that's quite a lot higher, because over the months after, so we recorded for a few months, it kind of leveled at around 5,000 clicks per hour per day and loggers. That's quite a bit less. So yes, echolocation did really go up when these things were there, when they were new, but after about 2 to 3 weeks, it goes down quite quick, and then it just kind of levels at a certain amount. So how did it happen that they, even though they already had the sea pods, the ones on the right, for a few weeks, how did they suddenly think these black sea pods were super interesting. We didn't know because we thought they look exactly the same, but let's put them in and see what they do. We did add some floaty devices so they wouldn't smash against the floor as much, and also these don't float as well. And somehow, even though to humans it's not very interesting, dolphins find this change in look really cool. So that was really nice that you can actually regain that those novelty behaviors by just changing the look of the device. So another thing that we found were the diel patterns. We want to see how high is their activity during the day. So the interesting thing is that most data that I found on other research is that they said dolphins were most active during the afternoon when the trainers are there, when there's people. But we found something really different. We actually found that after 6 o'clock, when everyone leaves, they all go to sleep, they start resting. There's hardly any echolocation recorded in that time. It's completely quiet. But then after 12 at night, around 2 o'clock or so, depend a little bit, could be 12, could be 3, they wake up and they start being super, super active, and there's a lot of echolocation recorded in that time. So I tried to see if the season maybe had anything to do with it, because in Sweden in summer it's really light out, and at 3 in the morning the sun already comes up. But even during the winter months when it was super dark, it was the same. The pattern was the same. So we didn't really find any difference in that. Looked at wild species. Very difficult to find anything on that, but I did read something on a wild group of bottlenose dolphins that forage a type of fish that's active at nighttime, and they forage around that time. But yeah, it's maybe a bit far off to relate that to these dolphins. So another thing that we found were different type of click trains and the behaviors that they display whilst showing these click trains. Mainly exploratory, exploring the devices, playing with the devices, or they swim past and they quickly move their head, like a bit like how you see it on the picture, whilst echolocating towards the device, to kind of use it like an orientation device, or maybe just thinking, oh, what is that again? Oh yeah, it's that thing. And this is what it looked like. So this is one that actually type of click train that you see when a dolphin is also approaching a prey, or when they're catching a prey. So the interclick interval is first very long because it's still quite far away. Then it locks onto the device, and you can see the click train getting shorter here, or the interclick interval getting shorter. It gets closer to the device. Here it's where you see the yellow line, it's a buzz. It's really quick clicks that you have there. It's right in front of it, and then it moves away again. So then the interclick interval becomes longer again, and then it swims off. So that's the type of click train that we saw quite often. And this is one that we saw often when they were playing with the device. So you see actually one dolphin here, and it's kind of like just smashing it around. And this is the kind of click train that you would commonly see then. So a lot of clicks emitted at that time, where here at the end it kind of moves away from the device again. So that's really interesting, really cool. And you can actually hear them when you sit there at the window. You can hear the sound quite well. So we also wanted to see if they use these devices for orientation, especially when it's night out, when they're resting, if they need these devices to, not bump into the walls, for example, to know where they are, or not bump into the devices themselves. But what we found was that when they're resting, they're actually really quiet. They're not really emitting any echolocation, or like sometimes a few clicks, but it's really, really not that much. And when I looked at devices to see if there was clicks recorded, and then I would look at the videos to see what they were doing, were they looking at the device or anything even going past it, but it wasn't really emitted towards the device. It was probably just kind of bouncing back again from the walls or something. So we couldn't really analyze this or conclude this hypothesis. So I went there at nighttime to see what was going on, if it was really dark, and how they even know how to swim around the pool. But there's a lot of exit lights around the entire pool, and dolphins their eyes are pretty much as sensitive as our eyes, and they can probably just see really well because the water is super clear, and I could see them without turning any lights on. So probably they don't need echolocation to not bump into anything. So the landmark use was the possibility for them to use it was most at night, because that's when the most echolocation was found. so that's why I went to the pool to see what was going on, and sat there for a few nights, the entire night, to see why so much echolocation was recorded during that time. But they rest, they make no sound, and then when they wake up, they're just super active. They're playing, and all the echolocation recorded during that time is actually because they're playing, they're fighting, they're socializing, they're doing whatever. They're super active. They're not really using it for orientation at that moment. So another thing we looked at is how interesting do they actually find the pool, or how do they move around the pool, and especially during rest, or not really rest, but more when nothing else is going on. If there's no trainers, if there's no play devices, if there's no training going on, see how they use the pool. And I made a beautiful picture, as you can see here. It's a typical path that the dolphin takes. They're always going with the clock around. They make a few circles in the deeper area, and then they kind of go back up again. So this whole corner is hardly used by the dolphins. Don't know why, but I don't know. They like to be here, and they like to be in the deeper area. And that's also what you can see in these graphs as well. So they really spend a lot of time in the deeper parts of the pool, the 6 meter deep parts. And they really like area 8. That's this area. Very popular area. That's because that area is right next to the kitchen. And they can actually spy hop here, and they can see into the kitchen, and they can see the trainers preparing their food, so they know when their food is coming. So they spend a lot of time there as well. There was actually one dolphin always there, always going around there. Yeah. So our main conclusions that we found. What I found most interesting is that they echolocate mostly after midnight and during the early morning, when there's nothing really, there's no trainers there, there's no toys or anything, and that's when most activity is recorded, which I found really interesting, because during the day there's a lot more activity, but somehow they don't use echolocation as much as they do at night. During rest, the possible use of using the devices as orientation devices is very low, and probably because they can just use their vision. So when we looked at enrichment with these devices, we saw that play and investigative behaviors decline very quick. Over 1 to 3 weeks, they hardly even look at the things anymore. But when you change the look, you put some floaty devices on it. So you change the acoustic target or make it look different, they can be super interesting again, and they behave the way they did when they were first introduced. Also, we found that season doesn't really have an effect on their activity patterns during the day. Also, we found that they spent more time in the deeper areas, and that more clicks were also recorded in the deeper areas of the pool, the 6 meter deep areas. And we also found, which a lot of other research also found, is that when they rest, they swim in a clockwise circular pattern, normally quite close to each other, in little groups. So how is this useful? Well, first of all, for the trainers, they really wanted to know, okay, what can we do to enrich these dolphins? What can we do to encourage their echolocation behavior? You can provide underwater structures, because we did find that it does encourage this behavior. But you do need to change them quick. You cannot keep them for too long. And that's a really important thing that a lot of zoos should remember, is that when you give them enrichment, you want to keep the purpose. So you want to take it away when it's still interesting. Because if you take it away when it's not interesting anymore, and you put it back a month later, they're like, oh, I know that thing already. I've played with that 1,000,000 times. It's boring. And then it loses its purpose, and you lose a lot of money and time spent on first getting the device. So you should take it away when it's still interesting, and then replace it or change it or whatever. So most zoos, the good zoos, they have an enrichment schedule where they constantly switch devices to keep its purpose. So that's really cool. And the trainers there, they actually started experimenting with these type of things, like constantly changing them, making like fake seaweed and things like that to stimulate their investigative and play behaviors, but also their echolocation behaviors. So that was it. And thank you for this opportunity. And I wonder if you have any questions. And I think I can probably look at the chat. I don't know what is best. Maybe there's already some questions. I'm not sure.
Can you hear me, Laura?
Yes, I can hear you.
Yeah, okay. Well, first, can I just, on behalf of everyone, just thank you very much.
Yeah,
it was really interesting. It's actually been a few years since I've seen that presentation, so it was nice to see it again. So thank you for that.
No
yeah,
problem.
I just also wanted to apologize to a couple of people who I think joined a little bit late. I think there was a problem with some passwords and signing in and things, so I apologize.
I think you recorded it, so they
yes,
can
yeah,
feedback, I guess.
we've recorded the webinar, so if anyone had any major problems, please let us know, and we can send you copies of that. But yeah, as Laura says, if you have any questions, maybe you can put a comment in the chat window. We'll see if anything comes.
Let's see, Andre has a question. One question.
Andre, are you there?
I see a question from Teresa. Thanks for sharing. How can we tell devices are no longer interesting? That's always very difficult, because you need to really do this properly. You need a researcher to sit there day and night and to constantly record behaviors and to say, okay, they're not really performing much behaviors towards the device. We should change it now. But of course, there's not much time to hire people like that. So normally people get students to do this kind of work for them. However, there's quite a lot of research on different species and how long enrichment should be in the cages. So for example, for tigers, they have these kind of protocols, or primates, probably dolphins as well. I would probably change it if you still see them being interested in it. That's probably best to maybe change it like every 2 days or something, and then make something completely different. Yeah. Okay, Luana says, pretty interesting. Okay. Did clicks decrease once trainers came in, or when visitors started showing? Not per se. It didn't decrease suddenly then. They actually became quite active again then. So you could see like a little peak, but then during the day it just kind of went less, and they started kind of resting. They become tired. The dolphins that were in the show, they had to perform, so they had to be active. But the dolphins in the pool that I was looking at, they kind of, they had like little breaks of high energy and then suddenly slow again, and they would rest. Yeah. So it didn't really have much to do with the trainers coming in, right? Although when the trainers left, that's when they all started resting. That was like their time to sleep, I guess. Were the first kids in the pool after something? Yeah, they were in the pool the entire time. The only time they were taken out was once a week, but they were only out for an hour, and then we'd put them straight back. So just take the SD cards out, put new ones straight back in, and then they would be back in the pool. Yeah.
Andre, did you have a question also?
Yes. Laura, can you hear me?
Yeah.
So one of, well, first of all, thank you for your presentation. That was quite interesting. And
No problem.
my question is about the conclusions of your work or your project. Yeah. So you mentioned in your first conclusion that the dolphins, they echolocate more after midnight than during the day. And do you manage to figure out why that's happened, or you have any idea why that could be happening?
Well, not really. That's what we found so interesting, because I didn't find any of that type of results from any other research, and there has been research done on activity of dolphins during the day, and they all said that the activity was highest during the day. So I was really interested in why that happened. That's why I spent a lot of time sitting next to the enclosure at nighttime, half asleep. But yeah, see what was going on. But they were just super active, and I think what I could also see is because they weren't really distracted. They didn't have any toys. There was no trainers. There was no food. There was a lot of fighting going on as well. So it could also just be echolocation for other purposes than orientation, maybe even for communication, possibly. Yeah, I'm not sure.
But the pool or the tank was dark by night and in the early morning, no?
It was dark, but there was these exit lights all around, and they were really light. So I didn't even have to turn on the lights to see the dolphins. I could just sit there and I could see them, and they're doing whatever. So it could be because it's a little bit dark, but we couldn't really find much proof of that. Because in Sweden at 3 in the morning in the summer, it's already light. So it could be really light at that time.
Okay. Yeah.
Yeah.
So they were not using echolocation just to see around because it was dark. They were using for
Yeah.
other.
Yeah, so it could actually be this behavior. We saw this in the winter time when it was really dark, but we saw the same behavior when it was already, like the sun was already out at that same time. So we didn't see any difference in that. So we're not sure why they use it more at that time.
Okay, thank you.
Yeah, no problem.
Laura, I think Teresa had one other question.
Okay.
Can you see that?
Let me see.
Do you reckon?
Oh, yeah. Do you reckon if the device removed most times maintain the pool for less time, maybe interest would be prolonged? Probably, yeah. So the best way, so this was just a research, we just wanted to see, how long these devices were interesting. So after 3 weeks, they weren't very interesting anymore, but we still were really interested in their orientation, type behaviors, and their activity around the pool. That's why we kept them for 5 months. But our advice to the trainers was to change such enrichment every week at least. It's difficult for the trainers to go and dive in the pool every other day, so that they can't really do that every day because it's also a little bit dangerous. The dolphins can pull out like the tubes or whatever. Only experienced trainers could do that, but we advise them to change it at least once a week, to keep their interest.
Okay. Laura, I had one sort of question. It's not so specific about the contents of your project, but it was just more regarding just conducting, research for a master's thesis. I know that some of our participants today are also biology students and might be working on projects of their own at the moment. And I know that you also, in your current job, working with students at the university there in the Netherlands to support them through their research projects. So I wondered if you just had any sort of a general advice or things. Obviously, we saw some of the problems that you had challenges to overcome in your project. So I wondered if you had any general advice for people.
Probably. I mean, a lot of the students that I have at the moment, they can be really creative and they can have these ideas. But I always tell them, look at other research, like read enough papers before you start trying things. See if anyone else did this before, if it worked or not, or if it might work for you. So read a lot of literature, and if it's a similar research, then just look at the methods. And it's okay to copy the methods. I mean, the methods are written down so that people can, do that research again or do something similar. So that's okay. So I always tell my students as well, look at the methods and try and use other research. You don't have to invent the wheel, a second time. So you can, that would be probably my best advice.
Yeah, so maybe don't sort of take on too big a challenge.
To just see little steps. And of course it's really cool to think of your own thing, like this with the sea pods in a dolphin pool that was never done before, but then you figure out these little things like, oh, okay, the data is way too noisy, we cannot read this. And luckily I had a really good researcher there which guided me, which is his name is Mats Amundin. Maybe you've seen his papers. He does a lot of research on porpoises. So he has a lot of experience. So get a good guide that knows about these things. And because I thought, oh, this is probably what the data is supposed to look like. And he was like, oh, this is unreadable. We need to change the settings. I don't know. So he told me how to do that. And so that's also important, to get a good guide that is involved with your project and knows about all these little things that you might not think of.
Yeah, a good supervisor.
Exactly.
Is, an important step. So I just wanted to check one more time, just if anyone has any final questions. We're almost, our time is almost up, but we still have a little bit of time if anyone has anything else they'd like to ask. Likewise, any questions, more general questions about research. Research projects. There's a question here from Luana. Laura. This is probably quite a tricky question.
Yeah, that is a tricky question. I think, you know, if the dolphins are already in the wild, you don't really have to encourage their natural behaviors because they're already using their natural behaviors. So that's quite a different way of looking at welfare, I guess. But I mean, it could be really interesting to see how much echolocation clicks a dolphin in the wild produces and compare it to dolphins in captivity. I mean, that would be really cool. I'm not sure how they would do that, but maybe with the devices these days you can.
Yeah, but
maybe.
Yeah, I think so. I think the second sort of part of Luana's question, she's saying, for example, looking at different vocalizations and seeing what they might mean, and that is something that
is really interesting. Yeah,
there's a lot of research going into that. So,
and I have actually the approach type click train that you saw, where at first the interclick interval is really long and then it becomes shorter. That is a click train that is also spotted in the wild as well, same as the buzz and all those other click trains, and the scanning one, for example. So that is quite similar, but I have written, I remember I wrote some research on the difference between echolocation and of dolphins in captivity and in the wild, and I do remember it saying that the frequency or the amount was different, but I can't really remember exactly, but there is definitely a difference in,
yeah. But yeah, as I say, there's certainly a lot of research, acoustic research, going into different species vocalizations and in the wild. We'll actually, the next week in the next webinar, we're going to look a little bit more generally at sort of acoustics and acoustic research for cetaceans. Obviously, Laura's project was a very sort of specific part of using acoustics in her research, but we'll look at that a little bit more generally next week. I think, is that we got any more last chance before we move on? I think it was good. There was some really good questions from you guys, so I'm glad to see that you all enjoyed Laura's presentation. If that's
Laura,
that's it. Oh,
yeah.
Sorry, can I just ask you another thing?
Of course.
Did you measure the noise that was produced by the filtration system, or did you try to check your results with the filter on and without the filter on?
No, we didn't do that. No.
Okay. Because
I think they did do a research on that before to see if their behavior was different with the filter on or off, but I'm not sure. We didn't do that in this research, no.
Okay, but during your experience, and the filter was always on, or it was on and off?
It was always on. Yeah, but
you
couldn't really hear it. I think it was quite a slight constant sound in the background.
Good, because that could be a problem for the dolphins, or one of the problems they have in captivity could be the constant noise that is coming out from the filters. But luckily nowadays we are aware of that, and we built more silent systems to don't disturb.
Actually, the system was actually in a different building, which was, I think, like almost maybe more than 10 meters away from the building. So the most noise was there, but that was quite far off, so they couldn't really hear that.
Yeah, the problem is the noise that could traveling on the water.
Yeah, exactly.
But okay, but the fact that you have the filter always on, I think it give you a more strong results.
Yeah, true.
Okay, thank you.
Okay.
Yeah, it's another good point from Andre as well, and it's another thing that we'll look at again next week. Sort of background noise, anthropogenic noise. Because this is obviously not just a problem or a potential problem for dolphins in zoos, but also for whales and dolphins in the wild. There's obviously a lot of anthropogenic noise
and then there's also actually with the dolphins in captivity that all the walls are concrete, so the echolocation sounds will bounce off these walls as well, so it will be quite a noisy environment, and that's what I remember research saying that's why their frequency is lower than in the wild, for example, because it's already so noisy.
Okay, that's interesting. Yeah. Sarah, excuse me, is asking if the filter systems could affect the behavior of the dolphins. I guess might be difficult for you to answer. As you said, the filter systems were always on, so
I find it difficult because they had I think they had quite a good filter system there, but I'm not sure. Probably in other zoos it's different, noisier. I can imagine that it will probably affect their behavior in some way.
Yeah, I mean,
but I'm not sure.
I think that sort of any any additional noise that the dolphins are exposed to will affect them in some way. It's difficult to say to what extent. And obviously, in Laura's project, she wasn't really looking at that.
No, we didn't take that in. So another question. I think you mentioned this, but wouldn't their own echolocation bounce back in a pool? Yeah, the echolocation does bounce back, sort of, in it's kind of, maybe not directly at them, but there will be like an echoing kind of noise probably in the pool from the concrete walls around. Yeah.
Okay. Okay, I think we are pretty close to our time. So, but that was really nice to see so many people with really good questions. I would, just like to, on behalf of Joanna and Andrea and the whole team at AIMM, and also everyone else viewing today, just really thank Laura for taking the time to do this. It's really cool to see you again, and we really appreciate.
Well, thank you. I really enjoyed it. So anytime.
So that's really cool. If you guys think of any other questions, anything that you would like to ask Laura, or indeed us, you can always drop us an email. You can see the email address at the bottom, hopefully at the bottom of my screen now. Before we go, I also just want to remind everyone again of our internship project. We've now started our season for this year. So if you would like to come and study these animals in the wild with us, learn about whales and dolphins, please drop us an email. Obviously, at the moment, I know that with the current situation, that traveling is not so easy for everybody. So if coming to Portugal is something that you're not able to do, we have a really exciting online course as well that we'll be running. This will actually be the second time we're running it. So it was a big success last month. So we've decided to run this again. So that will be starting on the 20th of July, so the 20th of this month. It's a 2-week course in 10 hours in total. Each day, or Monday to Friday, you'll receive like a video lecture. There's homeworks. There's also I think 4 hours of Zoom meetings. So we have Zoom meetings with everyone, and also an optional one-to-one session with one of our senior researchers here at AIMM. So it's a really unique opportunity. We had some fantastic feedback from our participants last month. So if any of you are interested in that as well, please do let us know. Also, for all of the webinar participants, we have an exclusive price for the online course as well. The normal price is €145, but for all of you guys, you have a huge discount, almost a third off, so it's just €100. So let us know if you're interested in the online course, or indeed if you're able to travel to Portugal, and you can come and actually study these animals in the field with us as well. Finally, just to remind you guys of next week's webinar on the 15th of July. It's next Wednesday, same time, so it's 4 o'clock in the afternoon, GMT +1. We'll be looking at acoustics and cetaceans, so a little bit of looking at acoustic research, but also some sort of mitigation measures, so where acoustics can be used to mitigate against some of the threats that cetaceans can face in the wild. So yeah, we really hope to see you guys again next week. I just want to say again, thank you to Laura. I see there's one or two other little questions that have come in, so we can actually keep the chat window open, and if you want to drop in.
I don't see the questions.
I think someone was asking what's next in your research.
Yeah, I typed the answer.
So you did. So you did.
My research career is over. No, I'm just kidding. No, I'm teaching now, so I'm helping other people get through their first research careers.
Very good, giving something back. So so just once again, thank you all for joining us. Thank you all for your donations as well. We really appreciate that, especially this year with all of the issues going on. So thanks again to everyone. Special thanks to Laura, and we hope to see you all again next week. So bye from Portugal. And bye from the Netherlands.